What's going on, friends? If you are joining us, there's like a little chat window that you can use in order to get in touch. And you can, of course, use the remind if you want. Don't be shy. I'm just going to stand here. Here's what I'm going to do. This is what I'm going to do for you right now because I'm just waiting for people to come on. I think I'm going to pull up a little respiration and photosynthesis, and let's just go over that. So I'm going to share my desktop right here. Boom. What's up? Hi, Mr. Kuhn. What's going on, pal? There's a little bit of a delay. I've noticed that there's like a 5-second delay, so don't be surprised if you write in and it takes about 5 seconds for me to get on board with you. But I was gonna just like put up my desktop and just go over some like photosynthesis and respiration to nobody except maybe to Bob. So here we go. Let's take a look. Pull these up. Nothing more fascinating than watching a guy search for files. Yeah, here we go. Right on. Can we full screen that bad boy? Oh yeah, we can. Oh yeah, we can. So the thing about photosynthesis that I think is really important to understand is that it's broken up into two major steps, right? We've got our light reactions, and then we've got our Calvin cycles. Let's go through the light reactions first. What's going to happen in light reactions is that we're going to use the energy from sunlight to split water molecules. Those water molecules are going to provide electrons, and when the energy from sunlight interacts with chlorophyll, that's going to cause chlorophyll to produce high energy electrons and put that into an electron transport chain. So we have this sunlight aspect going on at the leaves. The leaves are also going to be running the Calvin cycle, and we'll look at how these two things work together. So the light reactions are happening in the thylakoid membrane of the chloroplast, and we're going to use water and light and NADP+, and some ATP, some ADP, and then we're going to produce oxygen as a waste product along with some NADPH, and we're going to make ATP. So let's just talk a little bit about chloroplast anatomy so we can see how it works. If we look at a chloroplast, what we can see is we've got this double membrane structure, and inside of there we've even got another internal membrane, which is the thylakoid membrane, and that's where the light reactions are happening. So this is a blown up cartoon version of the thylakoid membrane, and what we can see here is we can see that light is interacting with the chlorophyll in photosystems. And so we usually start with photosystem 2. It's just the way that this gets done. So light interacts with the chlorophyll in photosystem 2, and that produces a high energy electron from a water molecule. This then goes through the electron transport chain. And of course, as that's happening, that's going to drive the process of chemiosmosis. So we have protons on one side of the membrane in the stroma, and we're going to pump those protons across the membrane from the stroma into the thylakoid space. So we're going to set up a gradient where we have more protons in the thylakoid space than we do in the stroma. Protons would energetically, we might say, prefer, although they don't really prefer anything, but energetically speaking, they don't like to be in one place. They all have this positive charge, they're being repelled from each other, but of course they can't go through the membrane. The only way that they can get back across the membrane is to go through a protein channel, and that protein channel is ATP synthase. And so we see that over here. We've got these protons going through ATP synthase. The energy that they release as they go through is used to drive the production of ATP through the phosphorylating mechanisms of ATP synthase, which you don't really have to worry about. If we follow the electron, so if we go back to photosystem 2 and we follow that electron's journey through the electron transport chain, it's going to wind up in photosystem 1. And when light hits photosystem 1, that electron is then going to be boosted up to a high energy level again. It can go one of two ways at this point. So it can go back into the electron transport chain. That's called cyclic photophosphorylation, and that will just drive the production of ATP. Or it can be used by an enzyme called NADP reductase to take NADP and attach a proton to it in order to generate NADPH. The bottom line is at the end of this process, we'll have ATP and we'll have NADPH, and we can use those in the Calvin cycle. The one thing that we'll just stress is that when the electrons wind up in NADPH, there's really no way for them to get back to the beginning. And so we need to put more electrons into photosystem 2, otherwise this whole process is going to shut down. And so the way that that works is that water provides those electrons, the water is broken apart, and it is then converted into oxygen gas and more protons. The oxygen gas leaves as a waste product, and that of course is why we can all be alive, because we're all aerobic cellular respirers. Let's go in and take a look at the Calvin cycle, pardon me. And remember that you can always get access to any of these slides whenever you want, right off of mrkanufke.net. They're all embedded in there. But let's go in and let's look at the Calvin cycle and the process of carbon fixation. So we have our ATP and we have our NADPH, and we're going to go and use those in carbon fixation in the Calvin cycle, which is going to happen in the stroma of the chloroplast. And so this is going to use carbon dioxide and NADPH and ATP, and that's going to produce G3P, NADP+, and ADP. Let's go back and just remember where we are. So the stroma is the space between the thylakoid and the outer membranes of the chloroplast, and that is where this process is occurring. So this is a very simplified picture of the Calvin cycle. We can start pretty much anywhere we want, but I usually like to start here with this blue enzyme, which is ribisco, or ribulose bisphosphate carboxylase, which of course no one ever wants to call it that, so we just call it ribisco. And the job of ribisco is to take carbon dioxide from the atmosphere and to smash that into the sort of end product of the cycle, which is RUBP, ribulose bisphosphate. That's going to then go through a series of complicated chemical reactions that you really do not need to worry about. But what you do need to understand is that the ATP is going to be used and the NADPH is going to be used to convert those RUBP molecules into 3 carbon molecules called glyceraldehyde 3-phosphate, also sometimes called PGAL, but I'm pretty sure that the AP exam is only ever going to call it G3P, or glyceraldehyde 3-phosphate. Some more ATP is used later on to turn those G3P molecules back into RUBP. But the bottom line here is that for every 3 times we do this, for every 3 molecules of carbon dioxide that we bring in, we're going to net 1 molecule of G3P that we can take out of the process. And this is really the end product of photosynthesis. I know we always talk about glucose as being the end product, but really it's this G3P molecule. There's a cool picture of ribisco. Yeah, but so here's G3P, and you can see these phosphates, and what this means is that it's really easy to smush G3Ps together into things like glucose. You would need 2 G3Ps, you could just smush them together. It's very easy energetically to do in order to make a glucose. Or plants can do all sorts of other biochemical pathways and turn this into amino acids, nucleic acids, fatty acids, all of the biological molecules that we will then get by eating plants or by eating other organisms that eat plants. So that's pretty much photosynthesis. Does that make sense? One other thing we can talk about here is that photosynthesis is really determining global productivity. And what we mean by that is that the amount of photosynthesis that takes place in a particular environment is determinant as to how much other living stuff can be in that environment, because producers, plants and photosynthetic prokaryotes and protists in the water, these are really the base of any ecosystem. And it's really how much energy that base is incorporating into the ecosystem that is going to determine the structure of the ecosystem that's built on top of it. If you don't have a lot of productivity, you really can't have a very complex ecosystem to build from. So I hope that that was helpful. Let's go in and let's see what's going on. Oh, I see that Jasmine is live streaming from during work. So that's cool. Jasmine, if you want, feel free to write in with any questions, and we'll be happy to talk about those questions. And I'm going to just bring it back to me. So apologies in advance, right? We'll just go right back to my ugly mug. So there I am. Yeah. All right, kiddies, if you got any questions, now is the time. If you don't, okay, but my heart goes out to you. Oh, what's that? You want me to talk about cellular respiration? Sure, I can talk about cellular respiration pretty easily. I'm going to do it a little bit differently here. So what I'm going to do is I'm going to draw it on a piece of paper as I talk about it. And then at the end, you can draw it too while I'm talking about it. And then at the end, we can put our pictures together and we can see if they look similar or not, right? So I'm going to start. And so the first part of respiration is glycolysis. So I'm going to start writing everything I know about glycolysis as I talk aloud as to what it is. So in glycolysis, what we have is we have glucose that we're going to start with, and we're going to break that glucose down into what? Do you guys know? Of course, we're going to break it down into 2 pyruvic acids, or 2 pyruvates. So that's really the major purpose of glycolysis more than anything else, is to take that glucose and break it down into 2 pyruvates. So if glucose has 6 carbons, then each of these pyruvates is going to have 3 carbons. Does that make sense? I hope it does. The other thing that we're going to get out of this is that we're going to get 4 ATP molecules. Now, we're going to have to use 2 ATP molecules to do that, but the fact that we get 4 out means that we've netted 2 ATP molecules, which is really important, because remember that glycolysis is one of these processes that is universal basically in every cell on the planet. It's one of only a handful of processes that seem to have been present in the common ancestor of all living things and has basically been passed on to all of the lineages of life. And that's because you can make chemical energy that the cell can use from this, right? For every 2 ATP that you use, you're going to get 4 ATP out. And so that's basically good enough for most of the cells on the planet, as long as those cells are not multicellular. The other thing that's going to happen in glycolysis is that we're going to wind up making 2 molecules of NADH. And that's kind of a big deal, because those NADHs, they're going to be used either later on in aerobic cellular respiration, or we're going to need to do something in order to turn them back into what they started with, which was NAD+. So what you see here is my glycolysis. I think that that is probably backwards, which is too bad, but that's how we learn not to do things like this. So yeah, if that was backwards, I apologize. That is certainly not the intent, but as you guys know, we're just kind of like learning by doing here as we go along. But I can certainly put up more of cellular respiration if you guys think that that would be helpful, or really do anything else that you guys want. It's your dollar, it's your hour, friends. So make the most of it if you would like to. We had a couple of questions about Hardy-Weinberg equilibrium, so we can talk a little bit about that as well. So let's see what happens if we do this. This may make a like weird, repetitive, like ongoing into infinity feedback loop, and so I apologize if that is the case. But I'm going to share my screen with my video on the screen, because I'm just trying things. I'm just figuring stuff out. Oh, I see. I can actually choose the content that I share. Well, that is super helpful. So here you can see we've got there's the slides from Evolution. And if we go and we take a look at our Hardy-Weinberg stuff, where is it in our slides? Ah, yes. No. Super important. I know you guys are psyched by this. Here we go. So Hardy-Weinberg equilibrium is going to be used to refer to a non-evolving population, a population that is not subject to any sort of evolutionary forces that are driving the population. And so we've talked about I think 4 or 5 different evolutionary forces. I usually focus on 4 of them, which would be natural selection. We've got genetic drift, sexual selection, and gene flow. And you really should be aware of each of these, what they are, and the examples that of each that you can use over the course of like the exam that you're going to be taking on Monday. But so in a non-evolving population, we're going to have all of these forces not acting. And so in order for that to be the case, we're going to need to have a large population size with no genetic drift. We're going to need to have the population totally randomly mating so that sexual selection is not playing a role. The environment is going to have to be completely stable, so that there's no natural selection. There's no immigration or emigration, right? No gene flow into or out of the population, and no mutation rate. So no real population is actually in Hardy-Weinberg equilibrium. It's just not something that is possible for a real population to be in, but we use it as a way to compare actual populations in order to see how evolution might be driving the effects of, or in order to see how evolution might be affecting that population's changing over time. And so in order to do that, we're going to use these 2 equations, and these are given to you on your formula sheet. So the first one is the allele frequencies, or the gene pool frequencies. You may see it talked about as the dominant allele and the recessive allele, usually the capital A or the lowercase a, or capital letter and lowercase letter. What we do in these equations is we symbolize P as the dominant allele and Q as the recessive allele. So we have our frequencies of P and Q, and when they add up, they both will add up to 1, because these are the only 2 possibilities that we can have. We can either be P or Q. Now, of course, individuals have 2 copies of each allele, and so individuals can either be homozygous dominant, in which case the frequency of homozygous dominant individuals would be the frequency of P times the frequency of P. Homozygous recessive, so that could be Q times the frequency of Q again. Or they could be heterozygote, which would be the frequency of P times the frequency of Q. But of course, there's 2 ways to get the frequency of P and the frequency of Q. We could get P from mom and Q from dad, or P from dad and Q from mom. So the total frequency of heterozygotes would be 2 times P times Q. So we have this genotype frequency equation that we use frequently as well. And so this is just showing you how they interact with each other in a perfectly Hardy-Weinberg equilibrium population. But the thing that I think is probably most important to understand about these equations is that the recessives are the key, right? It's always the recessives that are the key in these equations. You always want to find either the recessive individual frequency, or if you can get the recessive allele frequency directly, go right ahead. The problem with the dominant individuals is that the dominant condition, the dominant phenotype, is going to be shown both by homozygous dominant individuals and heterozygous individuals. And so as a result, we can't really know how much is going to be in each if we're just given that information. So even if we're given the number of individuals or the frequency of individuals that show the dominant phenotype, we'd still want to use that information to determine what the frequency of individuals were who showed the recessive phenotype before we did anything else. Because we can go right from the recessive phenotype frequency, Q squared, to the square root of Q squared, or Q, when we do this. The other thing to be conscious of here is that square roots in decimal land work really weird. So like if the frequency of Q is 60% or 0.6, then Q squared is going to be 0.36. And that's totally different than how we usually deal with frequencies, right? When we usually deal with frequencies, let me back up. That's totally different than how we usually deal with squares, right? Usually when we square something, it gets bigger, but here when we square it, it's going to get smaller. And so that's a really important thing just to kind of keep in your head. But once you figure out what Q is, then you can just subtract that from 1, and you can determine what P is. And then once we know what P and Q are, we can work our way through the entire equation. We can figure out the frequency of P squared, and we can figure out the frequency of Q squared, and we can do everything that we need to do in order to solve these problems. The most common mistake that we see is we see students that solve for the wrong thing. So they're asked to figure out the frequency of recessive individuals, and they solve for the frequency of the recessive allele. Or they're asked to figure out the frequency of homozygous dominant individuals, and they solve for both homozygous dominant and heterozygous individuals. So that is definitely something to be careful with and something to pay attention to. So I hope that that was helpful. And now let's go back to my beautiful face, and we can continue our conversation. So we got a comment in here from student, I suppose we'll say, about which evolutionary forces are random and which ones are not random. And I think that that's a really good thing to keep in your head. This notion of randomness and evolution is something that gets confusing. A lot of people sometimes think that evolution is totally random, and that's why some people have such a hard time really kind of accepting that evolution is a force that works on living things. And of course, there are some random aspects to evolution, but the thing to understand here is that natural selection is totally non-random. Natural selection refers to any time that the environment is determining which organisms survive and reproduce, which organisms are the most fit, which means essentially are able to survive long enough to have the most kids. And that is totally non-random. That's directed, right? It's being directed by the environment. Certainly the environment fluctuates, and that may cause natural selection in populations to oscillate back and forth. Like the classic example of this is Darwin's finches on the Galapagos Islands and the work of the Grants, right? We, of course, studied that, and we saw that the beak size would fluctuate, and it correlated to droughts in the environment. And it would go up and then it would go down, and we would probably oscillate back and forth unless the environment really started to become progressively drier in a more directed sense, which is certainly possible, but it's not what was happening in the example that we looked at there. But that's not random, right? The environment is determining which organisms survive and reproduce. And so as a result, those are the organisms that survive and reproduce. The other force in a population that is not random is sexual selection. If you are surviving and reproducing because you have traits that get you mates, then that is selecting for particular traits, and that is driving evolution in that population. And that's why we see things like hermit crabs with giant claws that are functionally useless, or peacocks with big tails that are functionally useless, because it does seem like these are the traits that the opposite sex wants in those animals. And so as a result, organisms that are produced that have those traits are the ones that survive and reproduce more frequently. But the other forces are random. So genetic drift is a classic example of totally random changes in a population over time. And there are a couple examples of genetic drift that we've looked at, but really it refers to any time in any situation where a population is just undergoing sort of random removal of individuals. So you could think about things that are totally non-selective, just like being unlucky. So like if you're growing on the side of a volcano when it erupts and you get wiped out, right, then you're not going to survive and reproduce. And so as a result, your alleles will be removed from that population. The other examples that we saw were things like the founder effect and the bottleneck effect. And in both of these cases, we had situations where a large population was reduced to a small number of individuals. And then that small number of individuals gave rise to a new population. And if you look at the allele frequencies in the descendant population from that initial small number, they're going to resemble the allele frequencies of that small population much more so than they'll resemble the population that that small population came from originally. We see this with reproductive isolation in certain populations of humans, for instance. The incidence of certain genetic diseases in populations of humans that have chosen to reproduce only among themselves can be at a higher rate, and that's because of these founder effects and these genetic drift effects that are happening. But we also can just see this just naturally in the environment. Generally speaking, the smaller the population is, the bigger an effect genetic drift is going to have on that population, because the more any one individual contributes to the overall gene pool. And so if you remove one individual from a small population, that's going to have a proportionally larger effect on the frequency of alleles in that gene pool than if you remove one person from a much larger population. So that's why when we go back to Hardy-Weinberg equations, the population needs to be really large in order for these random genetic drift effects to have no effect. So I hope that that was helpful, and thank you so much to the young man who wrote in with that question. Does anybody have any other questions? We're getting them off mostly of the YouTube chat window. You can just type your questions in there, and we'll be happy to handle them. We're also, of course, kiddies, you can get me in Remind if you want, and I'll get that at the phone. But yeah, I mean, it does look like we've kind of gotten our questions answered, and so assuming that that's all that we really need right now, I am going to sign off right after we talk a little bit about some exam prep stuff. So we still have our practice exam in our review book that we haven't touched. I would really recommend that you do that tomorrow. I think you should time yourself according to the time. The answers are at the end of the exam, and they are explained, and they are generally pretty good questions that are really going to model the questions that you're going to see on the exam. So I would do that tomorrow. I wouldn't do it on Sunday. As you guys know, the thing that you should do on Sunday is nothing related to AP Biology. If you really feel like you have to do something AP Biology related on Sunday, try to get it all done by noon. You really want to give yourself from about noon on Sunday until you wake up on Monday and go and sit for the exam. You really want that period of really not dealing with exam material. It turns out that if you spend a lot of time in that period, like cramming and trying to stick stuff into your brain, it doesn't work. And it can actually have a negative effect on the amount of information that you can retain over the course of the exam. So it's really important to give yourself that pause. A couple of other things for Sunday. You want to eat good meals, breakfast, lunch, and dinner, particularly dinner. Good meals, right? You really want something that's going to agree with you and that's going to give you nourishment. You want to get a full 8 hours of sleep. That is so crucial. It's really, really important. So I know that our school starts at 7:15, 7:22, right? So you would really want to be in bed probably no later, and I know that this is, but you would want to be in bed no later than probably 9:30, 10 o'clock in order to get that 8 hours of solid sleep. Obviously get to school on time, bring pencils and pens for the exam. You do not need to bring a calculator if you're my student, right? We will provide calculators for you. Certainly your teacher has told you if you need to bring calculators if you are you're somebody else's student. We're going to provide everything for you, and I'll see you there before we put you on the bus to take you over to take the exam. But other than that, I mean, that's basically it. If you guys have any last minute questions over the next 24 hours, 48 hours, you can always get in touch with me through our various communication channels. And I think probably the last thing that I'll tell you is that you should all believe that you can do well on the exam. It's really important to have a thought that you know this material and that you can actually demonstrate your understanding of the material. I'm certainly proud of you. I know that you know the material, and I'm excited to see how well you guys do on this test. So yeah, that's about it for today. Remember that we're doing the live general broadcast tomorrow, and that certainly you guys are welcome to attend that too, but I just anticipate that that is maybe going to be a little bit more attended than this one was. So I just wanted to give you guys the opportunity to get your questions answered here before we took it wide tomorrow. All right, thanks so much, guys. I really appreciate it. And yeah, have a great day.